If you are one of the millions of adults over 60 who starts every morning with a glass of plain water and feels good about it, this video is directly for you. Because right there in your kitchen, in that glass you reach for before coffee, before food, before anything else, your collagen synthesis machinery is waiting for the one nutrient it cannot function without. A nutrient that plain water does not contain. And the consequence of that daily omission has been accumulating in your skin since your mid-20s. Drinking plain water is not harmful. It is essential. But after 60, it is no longer sufficient on its own for what your skin needs. The single addition I am about to describe does something that plain water physically cannot. It activates the enzymes inside your skin cells that build the collagen framework responsible for firmness, smoothness, and structural integrity. Most adults over 60 are drinking the right thing. They are just drinking an incomplete version of it. That changes today. My name is Dr. Monty Fisher. This channel is built on one commitment: taking the published research on how the aging body works and translating it into information that is accurate, specific, and immediately actionable. Skin aging and collagen decline are areas where the science has become very precise. The molecular mechanisms are understood in detail. The cofactors that regulate collagen synthesis are identified and confirmed. And the research connecting nutritional support to those pathways is published in peer-reviewed journals. Today, I am going to give you that information in plain terms, because what you do tomorrow morning can be different from what you have been doing until now. Before the addition, you need to understand what is being lost, specifically, not generally, and why the timeline matters. Collagen is the most abundant structural protein in your body. In your skin, it makes up approximately 75% of the skin's dry weight. It is the physical scaffolding that keeps skin firm, smooth, and structurally intact. Type 1 and Type 3 collagen are the two primary forms in the dermis, the deep layer of skin where firmness and elasticity originate. From approximately your mid-20s, collagen production begins to fall. Dermatology research places this decline at approximately 1% per year. A study by Sibylla and colleagues, published in peer-reviewed dermatological literature, found that collagen content in the skin of participants aged 65 to 74 was approximately 25% lower than in those aged 25 to 34. That is, a quarter of the structural foundation of your skin gone before you reach your late 60s. For women, the decline accelerates sharply around menopause. Studies have found that women can lose up to 30% of their skin collagen in the 5 years immediately following menopause, a concentrated, steep loss that is added on top of the gradual annual decline. After that initial postmenopausal period, loss continues at approximately 2% per year for the following 2 decades. Dermal thickness, how thick and substantial the skin feels between your fingers, decreases by approximately 6% per decade. And it is not only production that falls. The collagen already present in your skin is being actively broken down by enzymes called matrix metalloproteinases, MMPs. These enzymes are activated by UV radiation, by oxidative stress from environmental pollution, by the advanced glycation end products formed when excess glucose binds to collagen fibers in a process called glycation, and by smoking. Research has identified distinct molecular transitions around ages 44 and 60, at which thousands of molecules involved in skin structure and integrity shift significantly, collagen-related molecules included. This is the daily reality inside the skin of every adult over 60, and most of it is happening silently, beneath the surface, years before the external changes become visible. Here is what plain water does and does not do. Plain water is essential for skin health. The dermis requires adequate hydration to maintain its structure, to support the transport of nutrients to fibroblasts, the cells that produce collagen, and to maintain the skin barrier. A chronically dehydrated dermis is thinner, more fragile, and less resilient. None of this is disputed. Water is the right thing to be drinking. But here is what plain water cannot do. The fibroblasts in your dermis need more than hydration to synthesize collagen. They need specific cofactors to run the enzymatic machinery of collagen assembly. Without those cofactors, the production process stalls at a critical step. The amino acid building blocks may be available. The fibroblasts may be present and functioning, but without the key that activates the collagen-building enzymes, the production process stalls at the hydroxylation step. The collagen chain cannot fold into its stable triple helix structure. It cannot form the structural fiber that your skin depends on. Plain water does not supply that key. The key is vitamin C. And what follows is exactly why, broken into 3 specific mechanisms, each with the published evidence behind it. Mechanism 1. Vitamin C is the non-negotiable cofactor for collagen assembly. Collagen synthesis follows a specific biological sequence inside your fibroblasts. After the amino acids that make up collagen, primarily glycine, proline, and lysine, are assembled into a chain called procollagen, a critical modification step must occur before that chain can fold into collagen's characteristic triple helix structure. That step is called hydroxylation. 2 enzymes, prolyl hydroxylase and lysyl hydroxylase, must attach hydroxyl groups to the proline and lysine residues in the procollagen chain, converting them to hydroxyproline and hydroxylysine. These modified forms are what allow the 3 procollagen chains to coil around each other, interlock into the triple helix, and form the stable collagen fiber that your dermis is built from. These 2 enzymes cannot complete this step without an active cofactor. That cofactor is vitamin C, ascorbic acid. Vitamin C acts as a reducing agent, maintaining the iron in the enzyme's active site in its functional reduced form. Without adequate vitamin C, prolyl and lysyl hydroxylase activity falls. The hydroxylation step fails. The procollagen chain cannot form the triple helix. The result is structurally incomplete, dysfunctional collagen that cannot hold the skin's architecture together. This is not speculative. This is the mechanism confirmed in the scientific literature as the basis of scurvy, the vitamin C deficiency disease in which connective tissue breaks down systemically. In scurvy, fibroblasts are present, amino acids are available, but without vitamin C, the collagen assembly line stops at the hydroxylation step, and the structural tissue throughout the body fails. You do not need clinical scurvy-level deficiency for this process to be impaired. Research published in peer-reviewed dermatological literature confirms that vitamin C levels in skin tissue specifically decline with age and are further reduced by UV exposure, environmental pollution, and oxidative stress, independent of total dietary intake. Meaning that adults over 60 can have adequate systemic vitamin C, yet still have suboptimal vitamin C concentrations at the skin cell level, where collagen synthesis actually occurs. Mechanism 2. Vitamin C signals fibroblasts to produce more collagen. The second mechanism goes beyond the enzyme cofactor role. Research published in PubMed-indexed literature has confirmed that vitamin C acts at a gene expression level. It upregulates the mRNA expression of type 1 and type 3 procollagen genes in human dermal fibroblasts. A study published in Biochemistry and Biophysics Research Communications confirmed that ascorbic acid enhanced the expression of type 1 and type 4 collagen and stimulated collagen gene expression in long-term human skin fibroblast cultures. In plain terms, vitamin C does not just enable the assembly process, It sends the signal for the fibroblast to produce more collagen in the first place. It acts both downstream, in the enzyme step where collagen is built, and upstream, in the gene expression step where the decision to build collagen is made. Laboratory studies have confirmed this. Human dermal fibroblasts treated with vitamin C produce significantly more collagen than fibroblasts cultured without it. Mechanism 3. Vitamin C protects existing collagen from destruction. The third mechanism operates through vitamin C's role as the skin's primary antioxidant. Your skin is exposed daily to reactive oxygen species, unstable molecules generated by UV radiation, air pollution, and normal metabolic processes. These free radicals do two things to collagen. They directly oxidize and fragment collagen fibers, and they activate the MMPs, those matrix metalloproteinases, that enzymatically degrade the collagen matrix. Vitamin C, as one of the skin's primary water-soluble antioxidants, directly scavenges these reactive oxygen species before they can interact with collagen fibers. It neutralizes the oxidative insult before the MMP activation cascade can begin. This means vitamin C is working in two directions simultaneously, supporting the synthesis of new collagen through the enzymatic and gene expression pathways, and protecting existing collagen from the oxidative degradation that is one of the primary drivers of skin aging after 60. The human evidence. A large cohort study published in the American Journal of Clinical Nutrition, Cosgrove and colleagues, 2007, examined the relationship between dietary nutrient intake and skin aging appearance in middle-aged women in the United States. After adjusting for age, sun exposure, menopausal status, and other dietary variables, higher dietary vitamin C intake was significantly associated with a lower likelihood of having a wrinkled appearance and skin dryness. The researchers concluded that dietary vitamin C was independently associated with a better skin aging appearance. This is observational data. It shows association, not proven causation. But the association is plausible, documented, and consistent with the well-established mechanistic evidence for vitamin C's essential role in collagen biochemistry. Most viewers who find something genuinely useful here move on without subscribing, and then they cannot find their way back when the next research-backed video comes out. If this video is giving you something specific and actionable, something you are going to do differently tomorrow morning, tap subscribe. 1 second, and you will not miss what follows. How to add vitamin C to your morning water. Option 1. Whole food approach. Lower dose, easy, and accessible. Squeeze the juice of half a fresh lemon into 8 to 12 ounces of warm or room temperature water. This provides approximately 15 to 20 milligrams of vitamin C. Drink it first thing in the morning before food, before coffee. This is the most accessible and lowest barrier version of this habit. Option 2. Targeted dose approach. Add 250 to 500 milligrams of vitamin C powder, ascorbic acid or buffered sodium ascorbate, to your morning water. This provides a concentrated dose consistent with the levels used in clinical research on vitamin C and skin outcomes. Dissolves easily in water, has a mild citrus-like taste, and is well tolerated at these doses by most adults. On timing. Morning is the most practical window for this habit. Before coffee, before food, before the day's competing demands arrive. Vitamin C absorption in the small intestine occurs primarily through SVCT1, the sodium-dependent vitamin C transporter 1, an active sodium-coupled mechanism. This transporter does not compete with glucose or food components the way some other nutrients do. The morning recommendation is primarily about establishing a consistent daily routine, which is what produces the sustained daily vitamin C availability that collagen synthesis requires. Taking it before the day begins means it is less likely to be skipped. On consistency. Vitamin C is water-soluble and is not stored in meaningful quantities by the body beyond tissue saturation. This means that daily, consistent supplementation, not occasional high-dose use, is what supports sustained collagen synthesis activity. The daily habit, not the one-time dose, is where the benefit builds. What to expect and when. Clinical research on vitamin C and skin outcomes typically observes measurable changes over 8 to 12 weeks of consistent daily supplementation. This is not a single-session result. It is a sustained biochemical process. The fibroblasts need time to produce new collagen, and new collagen fibers need time to integrate into the dermis. 4 weeks is an appropriate point at which some people begin to notice changes in skin texture and hydration. 8 to 12 weeks is the time frame consistently seen in published skin research. Vitamin C is the central addition to your morning water, but the collagen synthesis pathway has additional requirements that are worth knowing. Glycine and proline. These are the primary amino acids that make up collagen. They are found in bone broth, gelatin, and meat, particularly connective tissue cuts. If your diet is low in animal protein, or you rarely consume these foods, collagen peptide supplements provide a direct source of these amino acids. Zinc and copper. Both support collagen health, but through distinct roles. Copper is specifically required for lysyl oxidase, the enzyme responsible for cross-linking collagen fibers into stable, mature structures. Without adequate copper, cross-linking fails and collagen lacks structural integrity. Zinc supports collagen synthesis more broadly and plays a central role in regulating the matrix metalloproteinases that govern collagen turnover. Zinc is found in shellfish, pumpkin seeds, and legumes. Copper is found in nuts, seeds, and organ meats. Both are rarely deficient in adults eating a varied diet, but worth being aware of. Sun protection. UV radiation activates the MMPs that break down your existing collagen, regardless of how much vitamin C you are consuming. A broad-spectrum SPF, worn consistently, is the most direct intervention against ongoing collagen degradation from sun exposure. Reducing glycation. A high-sugar diet generates advanced glycation end products that cross-link collagen fibers, making them stiff and brown. Reducing refined sugar in the diet directly reduces this mechanism of collagen damage. Let me bring this together. After 60, collagen in your skin is declining at approximately 1% per year, a process that has been running since your mid-20s. By the late 60s, research shows approximately 25% of peak collagen content is gone. What makes this worse is that the existing collagen is simultaneously being degraded by MMPs, activated through UV, oxidative stress, and glycation. Plain water is essential, but plain water provides none of the vitamin C that collagen synthesis requires. Vitamin C is the non-negotiable cofactor for the two enzymes, prolyl hydroxylase and lysyl hydroxylase, that allow collagen's triple helix to form. Without adequate vitamin C at the skin cell level, these enzymes cannot complete their step. Additionally, vitamin C signals fibroblasts to increase collagen gene expression and acts as the skin's primary antioxidant defense against the oxidative degradation of existing collagen fibers. That is the Collagen Water Protocol, one addition to your morning water done consistently every morning. Step 1. 8 to 12 ounces of water, warm or room temperature, before food and before coffee. Step 2. Add your vitamin C, either the juice of half a fresh lemon for approximately 15 to 20 milligrams, or 250 to 500 milligrams of vitamin C powder, ascorbic acid or sodium ascorbate for a targeted dose. Step 3. Drink it first. Before anything else enters your system for the day, Absorption is most efficient before a full meal. Step 4. Make it daily. The collagen production benefit is cumulative, consistent, and builds over weeks. 8 to 12 weeks is the published evidence window for measurable skin outcomes. Step 5. Support the pathway. Adequate dietary protein for amino acids. Sun protection to stop ongoing collagen degradation. Reduced sugar to limit glycation. Tell me in the comments, have you been drinking plain water every morning without this addition? And are you making the change tomorrow? I read every comment. And if you know someone over 60 who talks about skin firmness, wrinkles, or collagen, share this with them. This is a 30-second change to a habit they are already doing. That is as low a barrier as it gets. Everything in this video is general educational information based on published research in biochemistry, dermatology, and nutritional science. It is not personal medical advice. If you have kidney disease, a history of kidney stones, or are on medications that affect calcium or oxalate metabolism, speak with your physician before regularly adding lemon juice or vitamin C supplements to your diet. High vitamin C intake may increase urinary oxalate in susceptible individuals. For most healthy adults, the doses described here are within well-established safe ranges. But your specific health history matters. This video is a starting point for an informed conversation with your doctor, not a replacement for one.